Hello, my name's Andrew Fisher. I'm Dean of Clinical Medicine at Newcastle University, and it gives me great pleasure to introduce to you the 2020 Jacobson Lecture. This is part of the Newcastle University Public Lecture Series. This lecture is being delivered by Professor Fiona Watt, and the title of her lecture is Understanding Stem Cells. We're delighted that Fiona Watt has been able to join us. Fiona is Director of the Centre for Stem Cells and Regenerative Medicine at King's College London and Executive Chair of the Medical Research Council. During her talk, she's going to discuss different types of stem cells in the body and why they're important both for normal function, but also in disease. I'd finally like to thank the Jacobson family for their continued support of our annual lecture series. Thank you.
Hello, everybody. I'd like to thank you very much for listening to my lecture today. It's a great honor to give this lecture, and I was really looking forward to coming to Newcastle and seeing my friends and colleagues. These are strange times, though, and so we have to make the best of the situation. So without further ado, I'm going to share my screen with you. Right, there we go. I like to start my talks with a picture of my lab, which is at the top of this little stumpy tower next to the Shard in the centre of London. And starting with this today, of course, is particularly poignant because I haven't been there in 2 months. So, as I said, these are strange times. I thought in this presentation today, I would tell you about my research on stem cells, but start off really with the broad background to stem cell research and what I see as the opportunities. I'll give you a little section of the presentation on my own research on skin and highlight a very fruitful collaboration that we have with Maslifa Hanifa in Newcastle. And then I'll tell you a little bit about the work of the Medical Research Council. I'm on secondment as executive chair there. And I thought you might be interested, because this is a lecture for the public, in some of the different ways in which we've responded to the COVID-19 pandemic and really done our bit to help the global research effort. So, what is a stem cell? You might be surprised to know that many articles have been written about this. It's been the subject of many debates. But for me, with my research background, it really starts with the question of how is it that as adults, the tissues in our body are maintained? And I believe that the origins of stem cell research lie in trying to answer that question, and particularly the interests of pathologists who would look at, under the microscope, at sections of healthy and diseased tissue and try to explore what was going on at the level of the cell. So if I go to the next slide, this illustrates what pathologists have seen for many years when they look down the microscope at a cross-section of human skin. We're actually looking here at the outer layer of the skin, which is called the epidermis. And each of these little pebble stones is the nucleus of a cell. So you can see that the epidermis is made up of layers and layers of cells. You can see that they change a bit as you move towards the surface of the skin. But most importantly, you can see that there are many dead cells. We know these cells are dead because they don't have a nucleus or any DNA, so they can't make more cells. And so people realize that as these dead cells are sloughed off the surface of the skin every day, they must somehow be replaced, because otherwise your epidermis would quickly wear away to nothing. And so this is one of the observations which led to the concept that stem cells are cells which are responsible for maintaining tissues in adult life. Another well-characterized example is the epidermis, is the blood. Oops. And I'm just going to go back into this. And in this diagram, you can see how the many different kinds of blood cells, which are illustrated here, all arise from a common founder stem cell over here. This name might be cryptic. It stands for long-term hematopoietic stem cell. And you'll be aware that there are different kinds of blood cells in the body. There are red blood cells or erythrocytes, platelets, T cells that fight infection. Some of these cells have a very short lifespan, and platelets are an example of essentially cell fragments that again have no nucleus and can't divide. So again, we have a stem cell which is responsible for maintaining all of the different kinds of cells in the blood. So what we can do with this sort of background is we can say that from the perspective of adult tissues, stem cells are cells that reproduce themselves, so they divide and make more cells. And some of these cells, that they're daughter cells, will undergo a process of further specialization, which we call differentiation. And that specialization might culminate in being a dead cell on the surface of the skin, or it might culminate in being a little red blood cell or a platelet. So this seems quite straightforward. I should point out that this is a picture which I've taken from Google Images and is definitely not an experiment in my lab. But it makes the point that when we're thinking about the tissues of our body, stem cells in blood make blood, stem cells in skin make skin, stem cells in muscle make muscle. We don't typically see any crossing of the responsibilities of the stem cells in the different tissues. But that leads into a completely different kind of stem cell, and that's called an embryonic stem cell. Now, actually, embryonic stem cells don't exist in the body. These are artificial cells which are made by isolating the cells from donated embryos discarded after in vitro fertilization procedure. And the cells from these embryos are grown in the lab. And when these cells are grown and expanded in the lab, they have the remarkable ability that they can be directed to differentiate into the different kinds of cells in the adult body. And because of this ability to make many different kinds of cells, these cells are referred to as being pluripotent. So what we can do is take embryonic stem cells in the lab, we can expose them to different culture conditions and growth factors, and then we can stimulate them to form the specialized cell type that are found in adult tissues. So just to illustrate that with a couple of pictures down the microscope, here is an example of a muscle cell which has been differentiated from an embryonic stem cell. And these little blue discs are nuclei, and you can see the little green stipples. And the green stipples represent the contractile machinery of the muscle. And another feature of muscle is that each muscle cell has more than one nucleus. So from an embryonic stem cell line, it's been possible to make a functional muscle cell. Here's another very nice example where embryonic stem cells have been directed in the culture dish to differentiate into neurons. Now, this is very interesting and contrasts with adult tissue stem cells. But things took, if you like, a rather strange turn over 10 years ago now, when this man, Shinya Yamanaka in Kyoto in Japan, showed that actually you could convert cells from adult human tissues into essentially embryonic stem cells by culturing the cells from adult tissues. It could be skin or bone marrow or nerve cells, and introducing into these cultured adult cells a combination of genes. These are the original 4 genes which Shinya used. And when you introduce this cocktail of genes, which are called reprogramming factors, you can convert all of these adult tissue cells into pluripotent stem cells. So these pluripotent stem cells have been induced in the dish, and that's why they're called iPS cells, to distinguish them from embryonic stem cells. Another characteristic of both adult tissue stem cells and pluripotent stem cells is that they don't just form individual shoots or collections of cells in a dish. If the culture conditions are right, they can actually be stimulated to form a mini organ, which we call an organoid. And these can be grown from adult tissue cells or from embryonic stem cells or induced pluripotent stem cells. And they can self-organize in three-dimensional cultures and show the ability to self-renew and differentiate as a mini organ. Here's a schematic of the way that you can take pluripotent stem cells, culture them in different ways, and then make them form what we call an intestinal organoid. So this represents the different cells that line your intestine. And in experiments performed in mice, these mini organoids have actually been used to repair a defect in the lining of the intestine. So this is really remarkable stuff. Here's a very nice picture from our colleagues at Cornell University, just illustrating the beauty of an intestinal organoid which has been grown in the dish. So I'm sure that you can see that there are a number of different possible avenues for exploiting these different kinds of cells that I've described. And in particular, induced pluripotent stem cells offer the tantalizing prospect that you could receive a transplant of your own stem cells to repair any particular tissue in any disease. This, of course, has captured the imagination of researchers, but of course, as you might imagine, there are a number of obstacles that have to be overcome to turning this into reality. So that leads me into the next section of my talk, where I just talk a little bit about stem cell therapies and other applications of stem cells. Now, I've just told you about the discovery of induced pluripotent stem cells, which led to Shinya Yamanaka's Nobel Prize in 2012. But actually, transplanting stem cells is a very old activity. And the best example of that is transplantation of stem cells from the blood. Now, blood transfusion is something which has been in routine use in hospitals in Britain for many, many years, long before we knew about embryonic stem cells. And it relies on collecting blood from donors in one part of the country and moving them, the blood cells, to wherever they're required for somebody who needs them, for example, after an operation or an accident. I'll tell you a little bit more about a more complex type of cell transplantation, which involves making human epidermis, the outer covering of the skin, in the lab and transplanting that. And that's just one further example. There are also ways of growing cells to repair the cornea or cartilage. Work with pluripotent stem cells is a bit further behind, but nonetheless, there have been important initial safety trials to evaluate the potential use of pluripotent stem cells to treat spinal cord injury and diseases of the retina. So lots of effort to make cell transplantation work to the benefit of patients. I'm just going to illustrate that again, going back to the outer layer of human skin, the epidermis, and show you this slide, which you've seen already. And it turns out that it's possible to recreate this cultured, epidermis in culture. And the man who really made this possible is the late Howard Green, who was a professor at Harvard. And he showed a long time ago, back in 1975, that you could grow cells from the epidermis. And he showed only 6 years after that first report that you could graft a burns patient with a graft of their own epidermis, which had been expanded in the lab. And this proof of concept from many, many years ago has stood the test of time. And this type of grafting of cultured epidermis is still in use today. This just shows you what it looks like. Once you've grown the epidermis in a culture dish, you need to detach it as a sheet before you transplant it onto patients. Excuse me, I'm just going to stop the dog barking. I doubt very much if we would have a barking dog if I was in a lecture theatre in real time. Sorry about that. So if I just go back, this shows you how when you've made a sheet of epidermis in a Petri dish, you can detach it. It's extremely thin. It looks, as you can see here, a bit like cling film you might use to wrap food. This is very, very carefully transplanted onto the damaged area of the skin. It's protected with a coating to stabilize it, sutured into place, and over time, this very thin sheet forms a durable epidermis. So how could we improve stem cell therapies? Well, there are a number of possibilities which are being actively explored, which I think will make this type of treatment mature. So one thing is to combine cell transplantation with gene therapy. And in gene therapy, what you're doing is you're correcting a defective gene. And a good example of that is the culturing cells from the epidermis of patients who have a skin disorder and transplanting this, correcting the gene, and then transplanting the corrected sheet onto the patient. The epidermis is a very simple structure, but if you think about the intestine or cartilage or types of muscle, you could imagine that to direct the cells into the right location and hold them in place, some sort of biomaterial or scaffold would be useful. And again, that's something that people are working on actively. Something that is very important to consider, even when you're transplanting somebody's cells back into the original patient, is that our bodies have evolved a very effective immune system to protect against damage and also protect against infection and recognizing foreign cells. So a lot of work needs to be done on how to control the immune response, avoiding a transplant from being rejected, and preventing other effects of immune rejection, such as scarring. This is a process sometimes called fibrosis. A fourth aspect, which is very fruitful to investigate and is the subject of a lot of work, is to think of ways in which you could stimulate the body's own cells to divide and differentiate. Because if you think about it, normally if you cut your skin, you don't need to have a transplant. Your body is able to heal the skin naturally. And finding ways in which the signals to stimulate the stem cells to divide and differentiate is another avenue that we can take. There are, however, some hurdles which are listed on this slide, which we have to overcome even if we have a treatment that really works. And of course, one of the big questions is how can this treatment be delivered within the context of our National Health Service? How much will a treatment cost, and how many people can be treated, and what is the benefit in terms of quality of life? Another aspect, which of course has to be considered, is that where you have treatments, whether they're drugs or cell therapies, there's going to be some aspect of commercialization. And when it comes to stem cell therapies, there is a very complicated patent landscape that needs to be considered. Then you also need money if for commercialization. And I think it's probably still true to say that it's easier to raise money in the US than in Britain, although we are seeing venture capital firms investing in Britain. And certainly American investors look at British science very carefully and favorably. There aren't very many companies in this business at present, and by and large, I think it would be fair to say that large pharmaceutical companies are risk averse. These technologies that we're describing are quite new, and so often the innovation will take place in a little biotech company, and when things take off, it will be taken over into a larger company. So all of these points I've listed are serious obstacles to getting cell therapies into the clinic. But in green, I've listed two major opportunities which are well appreciated. One thing is that in recent years, different types of immune-based therapy for cancer have been highly successful. And many of those involve actually a combination of cell and gene therapy. And because these have been not only successful in treating certain types of cancer, but they've also been commercially successful, this provides large and small companies and scientists a guide to the possibilities of making stem cell therapies part of the clinical, routine clinical practice. And another thing is that we don't have to think simply about commercializing cell therapies. We can think about how little companies might make money from devices or scaffolds or the materials that you need to expand cells in culture. So it is a difficult field, but I'm very optimistic that things are moving forward in a positive way. It's not just about transplanting cells into the body, of course. You can use stem cells very effectively for disease modeling and drug discovery. And I'm going to, in the next couple of slides, just illustrate what I mean by that. So here's a diagram, and the different steps in this should be familiar to you. It shows you over here on the left-hand side how you can take cells from a patient, so cells from adult tissues, reprogram them with those Yamanaka factors to convert them into induced pluripotent stem cells, and then you can direct them in the lab to differentiate into different cell types. Once you have these cells in Petri dishes, you have a number of opportunities. So as I'll illustrate in a minute, you can use them for modeling complicated diseases. You can also use them for drug screening. And this is quite important because although the step for getting drugs approved to patients will generally involve experiments in animals, we know that drugs can fail if it turns out that their ability to interact with human cells is different from cells, for example, in a mouse. So if you can have better disease models and better human screens for drug discovery, then you can increase the likelihood of success once you move these drugs further. And something else that is really important is that whenever you're developing a drug to go into a patient, you have to be aware of potential side effects. And those will often be affecting the heart or the liver or the nervous system. And of course, if you can, in a dish, form heart cells, nerve cells, and liver cells, then you have the ability to screen out drugs which might look encouraging, but would have very unwanted side effects. Now, I'm just going to illustrate disease modeling with some experiments that are going on in my own lab at the moment. And they arise out of a very ambitious project which we started 8 years ago, called the Human Induced Pluripotent Stem Cell Initiative, or HIPSI as it's affectionately known. What we did in this effort, which was involved a number of labs around the country, was to make iPS cells from a number of different donors. And what we wanted to do was see if we could do some experiments relating the genetic background of the cells to the phenotype, if you like, of the cells in the patients and the disease. And I was really privileged as part of this to start a very fruitful collaboration with Professor Philip Beetle at the Institute of Child Health. And Phil is world-renowned for his work with patients who suffer from a disease called Bardet-Biedl syndrome. This is a fascinating disease. Fortunately, it's very rare. But what is puzzling is the different kinds of symptoms that patients have. These symptoms are listed here. Unfortunately, patients tend to go blind over time, and other aspects of the disease can be more or less prominent. For example, quite commonly, patients will be obese. One strange phenomenon of the disease is that they will often have extra fingers or toes. So you can see here, this child has got 6 toes instead of 5. You see other conditions, and the one which caught our attention was that you see defects in kidneys. And often these patients with Bardet-Biedl syndrome will manifest with kidney problems when they're young adults. So we've got a rare disease, we've got multiple phenotypes, if you like, and we know that there are 17 different genes which, when they're mutated, can contribute to this. So what we decided to do was to team up with Phil's group and make kidney organoids from different patients who had mutations in the same gene but had different degrees of severity of their kidney defects, and see if we could get any clues about what might be going on. And so this shows you how we can grow kidney organoids in the lab. It's a process which takes about 3 weeks, and we're looking down the microscope at what the kidney organoids look like. Now, if there's anybody in the audience who is a nephrologist or has actually even eaten kidneys for lunch, you will see that this is not by any stretch of the imagination a kidney. It's very, very small. But if you zoom in, you can see within the organoid little duct-like structures. And these elements contain the differentiated cells that we see in kidneys. So we can recapitulate at least some aspects of the kidney using this protocol. And so what we did with Phil was to simply have a look at kidney organoids formed from patients who have the same mutations in the same gene, BBS10. So this is an organoid, kidney organoid from a healthy patient. And then these are kidney organoids from 3 different Bardet-Biedl syndrome patients. And interestingly enough, although the same gene is affected in these 3 patients, this patient had a much more severe kidney problem than the other 2. And you can see that we've recapitulated that in the culture dish because this structure has not formed properly at all compared to the others. And so my students, Chloe Hurley and James Williams, have done a lot of work on developing this protocol. And James has now made great strides in working out not only why there are kidney defects in BBS10 patients, but why some are more severe than others. Now, before I switch to telling you about the work of the Medical Research Council, I'm going to wrap up the stem cell research part of this presentation by telling you a little bit more about the work that we do on skin. So you'll have gathered that my lab is very interested in the outer covering of the skin, the epidermis, and we've done a lot of work over the years with cultured sheets of epidermis that I've shown you already. And so our work has really been transformed in the last 3 or 4 years by a global initiative called the Human Cell Atlas. And there are many participants in this around the world, but it is led by Aviv Regev in Boston and Sarah Teichmann at the Wellcome Trust. And the aim here is essentially to do Google Maps for the human body, to be able to say for any given tissue what all of the cell types are in that tissue, and also how they're arranged in space and time. This is a really great opportunity to get new information about how the body works, so normal human physiology, and also to start to piece together disease mechanisms. So there are potential practical benefits from the Human Cell Atlas, but in addition, the Atlas is driving completely new ways to integrate different kinds of data, to foster collaboration and open data sharing. So I've been absolutely delighted to be part of it, and in particular, to be working on the different cell types in human skin. So I suppose in a way, up till now, I've cheated a little bit because I've explained how simple human skin is. But actually, if you look at this cartoon, it illustrates just how complicated it is in fact. We've got the epidermis, which I've described. It's supported by a connective tissue. We also have hair follicles, sweat glands, sebaceous glands. We've got blood vessels, we've got fat, we've got nerve cells. So trying to understand what all of these cells are and what they're doing is quite a challenge. And in this project that I'm involved in, we collaborate with Maz Hanifa from Newcastle, Sarah from the Sanger Institute, and then we have great collaborators, including my team at King's College London. Now, I'm not going to take you into the details of how we map the cell types in the skin, but essentially what Maz and the team have done is to take biopsies of skin from healthy donors or patients, for example, with psoriasis, disaggregate the tissue into single cells, isolate the RNA from the cells, and then have a look at which genes are expressed. And through complicated bioinformatic analysis, we essentially come up with the map that you can see here, which is color-coded just to indicate the different kinds of cell populations that we can find. And this has been hugely helpful because we can integrate the data into other types of analysis to really get a much richer picture of what's going on in the skin. I'm particularly keen on using the Cell Atlas to help us understand data on what we get from if we image the skin using a non-invasive method, which is illustrated here, called optical coherence tomography. Essentially, it's very like the sort of ultrasound that you would do to have a look at the baby growing in the womb. But the probe is run over the surface of the skin, and very quickly you get information about the different layers of the skin. So here in this moving cartoon, you can see the skin surface there. It's being tilted here. Then we can zoom down into the deep layers of the skin. I should emphasize completely painlessly, that's the epidermis, that's the connective tissue. And these dark streaks here represent blood vessels. Now, one thing that we can do very easily is map the pattern of the blood vessels, which is shown here in red. And when we do that, we can ask questions about the nature of the blood vessels in different body sites and ask whether any variation that we see in the blood vessels correlates with any variation in the map that we get from the single cell profiling. So this is very much work in progress, but I thought I'd share a slide from Clarisse Garnier in the lab who's working on the project. And what Clarisse has done here is to simply compare the pattern of blood vessels in different body sites. So the stomach, forearm, the back, forearm, palm, and other sites such as the nose and lip. And I think you can see very clearly that the pattern of the blood vessels is quite different in different body sites. We're seeing a very high density of blood vessels in this sample of palm compared to a low density in the forearm. And we're now trying to integrate that with the single cell atlas data to ask questions about the nature and the function of the blood vessels in these sites. Another practical application of the human cell atlas work, which is one that our lab is very involved in, is asking whether we can use the information we have about cell types in the skin for developing new treatments that might be beneficial to resolve long-term scars, such as this one, which is illustrated here. We know from the human cell atlas that there are multiple different kinds of fibroblasts, and our studies in the lab suggest that one particular population of fibroblasts, if it was injected into a scar like this, would be very beneficial for helping the scar to resolve. And of course, when we think about scars, we're not simply thinking about a cosmetic problem. We're thinking about scarring which can greatly restrict the mobility of the joints. So this, as I say, is work in progress, but it has been greatly accelerated by the collaboration that we have through the Human Cell Atlas. So now I'm going to move to the last part of my talk, and I'm going to tell you a little bit about the Medical Research Council. I feel privileged to run the Medical Research Council because we fund fantastic biomedical research, and it's a great responsibility because the money that we distribute comes from the taxpayer. So we're very conscious that this money is money that is not being spent on other things, and we're really mindful that it should be of benefit to the whole population and indeed the world. So the mission of the Medical Research Council is to fund research that will improve human health. And actually, we've been going strong since 1913. And so it's interesting, you will know that many people are keen on rebranding products and services every few years. And actually, the MRC mission hasn't required a rebrand in all of that time. If you're interested in what we fund and our plans for the future, there's a lot of information on the website. But here I've just captured a summary from the delivery plan that we published last year, because it provides quite an interesting timeline from 1913 through to 2018, we're just picking up one or two of the discoveries. So, for example, the MRC was set up to tackle tuberculosis. Scientists went on to discover different kinds of diabetes, to show a correlation between inactivity and increased risk of heart disease. Researchers also found that exposure to asbestos increases lung disease, and there are many other discoveries. For example, the developing humanized monoclonal antibodies has led to many of the advances in cancer immunotherapy to the present day. And just right up to 2 years ago, and work that is carrying on at the LNB in Cambridge now, is essentially artificial biology, engineering cells in completely artificial ways to make molecules that they wouldn't do normally. And although this could be classified as blue skies research, we think it is going to open many new opportunities for developing new types of drugs to treat a disease more effectively. In our delivery plan that we published last year, you can see some of our priorities. I always think this looks a bit like an old-fashioned record, but they've come into fashion in recent years, so I make no apologies for it. But at the heart of what we do, we want to invest in people. We want to make sure that we're funding the best discovery science that we can. We want to encourage scientists to collaborate with one another and also to work in partnership with members of the public and patients who will give us very good insights into the kind of approaches that we should be taking to work on particular diseases. And of course, in medical research, technologies and infrastructure are changing, and requirements become more and more expensive. So we want to make sure that when we're investing in people, they have the technologies that they need to succeed. 10 years ago, if we were talking about what the MIC does, we would probably have drawn a line from the laboratory or the bench to the bedside. But I think that linear progression is rather outdated. And so in our delivery plan, we've put partnerships for health and economic impact right here in the center. So this is collaboration with clinicians, nurses and social workers, for example, and also making sure that when it comes to issues like commercialization, we're ready to go. And you can just see some of the things that we believe to be really important. I might highlight, for example, mental health, because we have a big effort at the moment in adolescent mental health, and something here called multimorbidities, which is essentially recognizing that particularly as you get older, you may not just have one disease. You may have two diseases or more. And the trouble is that to date, treatments tend to be focused on a single disease. And as I'm sure many of you will be aware, sometimes patients can end up taking multiple drugs, which don't always work very well with one another. So this issue of understanding multimorbidities and dealing with it is very important for us going forward. So that's really what the MIC does, our overall priorities. But then in this slide, I've illustrated some examples of the kinds of things that we invest in. So I've already mentioned the MIC Laboratory for Molecular Biology in Cambridge. It's this rather nice building here. And from this discovery science has spawned a number of Nobel Prizes over the years, and also some really important applications that have been very important for human health. A different kind of investment is UK Biobank. This is a partnership with other funders and started about 15 years ago now to recruit a cohort of 500,000 people who altruistically agreed to provide a sample and then agreed to be recalled for other sorts of analysis. And as we've gained more and more information about how different genes contribute to disease risk, the biobank resource has become more and more valuable. And we see really interesting information coming from imaging studies, for example. Another example here is the Dementia Research Institute. I give this as an example because although we call it an institute, it's not one physical entity like the LMB, and the network of researchers are based in different parts of Britain and coordinated by a central hub at UCL, but with partners, for example, in Cardiff and Edinburgh and elsewhere. Another kind of partnership that we have is this one called the UK Prevention Research Partnership. The logo is down here, and you can just see how we're working with multiple funders to do some really innovative research. And I believe that one of our very successful partnerships has a component here in Newcastle. The last example on this slide is our MIC unit in the Gambia, and I've illustrated with this photograph here from the website. Now, Gambia is a really, really small country, and our unit has been active there for many years. So the relationship between our researchers and the population in Gambia is really extraordinarily close. And our clinicians provide actually quite a lot of frontline care. So what we have is a wonderful opportunity for researchers from Britain to go and be trained in the Gambia, contribute to healthcare there, but also get exposure to a lot of diseases and problems that they wouldn't see so readily in Britain. So these are the examples of types of MRC investment. And then I just want to end on how the MRC has responded to this global pandemic that is COVID-19. Now, back in January, it was clear that we were going to have to do something to help with the global effort to deal with this disease. And I contacted Chris Whitty, who many of you will be aware of as Chief Medical Officer. And I said, I think we really ought to do something as a partnership. And so what we did in close collaboration with the Department of Health and Social Care was decide on rapid funding that needed to be carried out. We started off with two calls, which were, one was for vaccines and one was for other aspects of COVID. And then this has morphed into the rolling call, which you'll be aware of, well, some of you may be aware of. What is really impressive about that is that one of the projects that we funded was for initial vaccine workup in Oxford. And just a week or so ago, Oxford announced that this vaccine trial is moving into phase 2 and 3. And in fact, a big partnership with pharmaceutical company has been announced. So this has been a very new way for us to work. As I said, it's a close partnership. And based on that, within other elements of the research landscape within UKRI, there are rolling calls for funding going on. But we weren't starting from scratch, and other investments that you would be aware of are money that we were able to give to our existing investments. So I've mentioned institutes and units. We also have research centres, and some of you will have seen the work of Neil Ferguson at Imperial College, who runs one of our centres. We knew that Neil had expertise in modelling pandemics, and his advice to government has been really crucial from the get-go. And then going back to our unit in the Gambia, what they needed was to make sure that they could protect their frontline workers for any cases that would come in and be ready to help. And I follow them on Twitter, and I just did this screenshot from the 19th of May, where they give a daily summary of their COVID-19 testing. Now, you'll be aware that in Britain, we're trying to do 100,000 tests a day. The scale of testing in the Gambia is quite small, luckily, but it just shows you the importance of this kind of investment. And then the last example that I'm going to give here is something which has launched, which is called the COVID-19 Protein Portal. I like to describe this as Amazon Prime for scientists, because essentially researchers in many places, for example, in our unit that we fund in Dundee and our virus research centre in Glasgow, have a lot of expertise in studying coronavirus, and they're very good at making pure proteins from the virus. So essentially what they did was say, we're very good at making proteins, and we will provide them for researchers who would like them to speed up the development of antibodies, for example, to improve diagnosis or potentially for different kinds of treatment. And so this is a wonderful example of how researchers have come together to collaborate to work on this disease. I suppose the point that I want to make overall about this is that We've seen the importance of science and research in shaping the response to the pandemic and hopefully getting us out of it. And I just want to emphasize that we were not starting from scratch. So as I've mentioned, Neil had a lot of experience in modeling, and the group in Oxford who've been so quick at developing the vaccine already had experience of vaccine development for other diseases. And if we think about how long it took to get from the identification of a target to a first in human study for SARS back in 2003, I think that was of the order of 20 months. And if you look at how the world has responded to vaccine development for COVID-19, we've shrunk from 20 months to 65 days. And that really is remarkable. And even though we're in a difficult situation and many lives have been lost, you can be sure that we're learning all the time about how to work faster, more collaboratively, and really do our best to make sure that we cater to the diverse needs of the community. So that's all that I wanted to present. Once again, I'm really sad that I'm not with you in person, and I will rectify that as soon as we're allowed to travel. And I just want to end with a slide which goes back to the research that I described on stem cells. So this is taken at our lab retreat, and it really makes the point that our researchers truly are superheroes, not just in my lab, but in labs all over the world. And I'd like to thank you for listening, and I'd be really happy to take questions. So thank you very much.